All right, students, so in this module, we're going to talk about, how does DNA contain the instructions for a protein? In the previous lesson, we mentioned that DNA is instruction for how to build a protein. We mentioned that protein is made up of amino acids. It's the building blocks of a protein. And so in this one, we're going to look at the genetic material. So DNA is the genetic material. And it's important because everything that happens inside your cell uses a protein, usually. Your muscle cells contract because of proteins. You can send signals from your brain via hormones because of proteins. They're kind of the workhorses of the cell. And the instructions for all of those proteins are contained within the DNA. And it wasn't until the 20th century, really, that we began to perform experiments that determined the nature of genetic material. And this is one of the most famous experiments. It's Frederick Griffith's experiment. And what he did was he took pneumonia strains, and he was experimenting with, what is it about these pneumonia that kill the mice. And if you look at this pneumonia strain here, it has a capsule around it. And this capsule protects the pneumonia and it keeps the immune system from attacking it. The capsule itself is made of protein. So he injected live S strain, or for smooth strain, into the mice. The mice died. If you get pneumonia without the capsule, they look rough. That's why they're called R strain. Injected those, they didn't die. If he injected heat-killed S strain, so he wanted to see if the capsule itself was the thing causing the mice to die. If he injected that, they didn't die. But if you heat-killed the S strain, which didn't kill mice, and the rough strain, it didn't kill mice. If you injected these two things together into the mouse would still die. And what would happen is if you took a blood sample, you would find living S strains, which means that the rough strain picked up the instructions for how to build the capsule from these bacteria and could cause the disease. Scientists, Oswald Avery added the enzyme DNase to Griffith's experiment. It breaks down DNA molecules, and when DNase was present, the R strain did not occur, and it suggested that DNA was the genetic material. Hershey and Chase conducted an experiment with a virus called a bacteriophage, and by tracking that bacteriophage, they ended up identifying that DNA, not protein, is the genetic material in cells. And this was the Hershey and Chase experiment. In the 1940s, this guy named Erwin Chargaff identified the pattern of nucleotide base pairing. So if you look at, DNA has two strands. This strand here we call the backbone. And between the backbone are the nucleotides, the A's, the T's, the C's, and the G's. And he noted that for every A, you had a T. So these ratios were 1-to-1. And so he noted that, and he noted that the ratio, the percent of guanine is always equal to cytosine. So he correctly presumed that these two things were paired together in some way. Then chemist Rosalind Franklin used X-ray crystallography. What you do is you freeze DNA, you crystallize it, and then you shoot an X-ray beam into this crystallized DNA, and it will diffract some pattern. And because of this pattern that we see here, this X-like pattern, she realized that DNA was a double helix. And because of, Chargaff's ratio, Rosalind Franklin's work, Watson and Crick put all the former findings together to build a model of the double helix molecule, and so they figured out the structure of DNA. And this is the double helix here. This is the backbone. The backbone is made of phosphates and sugars, and in between are these nucleotides, the A's, the T's, the C's, and the G's. So how's DNA copied? And so now that we know what it looks like, your body has about 30 trillion cells in it, but all of them share all of the genetic material. And all the time in your body, somewhere those cells are dividing. And your cells have 46 chromosomes. You get 23 from your mom and 23 from your dad. And if this cell divides into two new cells, each new cell also has to have 46 chromosomes. So 46 for that one and 46 for that one. Well, you can't take 46 and make two new 46s, and so DNA has to be copied. And that, that information is copied through what we call replication. And DNA is replicated semi-conservatively, which means that if you have a parent strand, so this is the parent DNA, what will happen is these two strands will separate and two brand new strands will be built between them. So eventually this, oh, that was terrible. Eventually this will grow. I can't draw a straight line with the mouse, sorry. But you'll have two new strands of DNA, and each one will have a parental strand or an old strand. And this, here's a much better drawing of it. DNA is being copied here. The double helix, as you can see here, has been separated. The two strands have been pulled apart, and a new strand will be copied from the old strand. And the reason that we can copy it is because, look, there's a G here, and a C always goes with G. And the enzyme that replicates your DNA can recognize that there is a G here, and it knows there's a G there, and so it knows that a C belongs with it. And if you look over here, here's a C by itself, and of course we know that a G will be coming next. And by doing so, if you look, you end up with two exact copies of one another. Well, the process of DNA replication occurs in several steps, and one of the first steps is this thing here. Oh, excuse me. This thing called helicase. It's the job of helicase to take the parental strand that's bonded together and basically unzip it. So we see part of the parental strand going this way and part of the parental strand going this way. Another important player is this thing called DNA polymerase. What DNA polymerase does is it recognizes unpaired nucleotides, like here. These are unpaired, and it pairs them up. So you'll have that going on this strand this way, and because DNA polymerase can only work in one direction, this polymerase will work this way, but it does so in kind of a loop. It will copy, so it will copy this way, it'll keep copying, and once it reaches this previous copy, it will unattach, it'll move back up, and it will reattach here where it will copy again. And helicase keeps moving this way. Because DNA polymerase works even outside the cell, we can get it to work in a test tube. We have this pretty cool technology called PCR, or polymerase chain reaction, where we heat DNA up to about 95 degrees Celsius, And if you heat it up that hot, it becomes single-stranded. And then we don't need helicase in that sense because now we've taken DNA and we've made it single-stranded. Then we cool it down to about 50 to 60 degrees where we can get some primer to bind. And a primer is a landing place for these polymerases. If you look, there is a primer here, here, and this DNA polymerase can only bind to a primer, so we have to have one. And then extension at 72 degrees where polymerase adds complementary bases to each of the single DNA strands. So this is what happens here. You heat it up, DNA pulls apart, cool it down, the primer will attach, then you run it at 72 degrees and you can elongate. And so even if you start with 1, you end up with 2. And at the end of cycle 2, you have 4 copies. At the end of cycle 3, you'll have 8. At the end of cycle 4, you'll have 16, and it doubles every time. So what is gene expression? How do we get from a gene to a protein? So a gene, of course, is a segment of DNA that contains the instructions. Gene expression is the series of events within a cell that take that information and produces a protein from it. Well, the information in the DNA and proteins are written in different languages. DNA is written in a series of nucleotide bases, the A's, the C's, the G's, and the T's, while proteins are constructed of amino acids. And DNA and protein machinery are located in different places. And so there's a problem. And I like to use the analogy of a library. And so the library is like your nucleus. And inside the library, you have all these instruction manuals for how to build stuff like your genes and your DNA. Now, you, as someone who lives in this town, the cell, need to build a new church or a new library or something. And the instructions for how to build the library, let's not use the library. The instructions for how to build a movie theater are inside the library. Well, you go into the library, you find the instructions for how to build the movie theater, and the library will not let you check the book out. The book is precious. It's the only copy they have. They will not let you take it out. But what you can do is make a copy of it. And so you make a copy of it, you carry the copy out of the main library, and you go to the work site, and then you assemble all the parts together to make that movie theater. So what we're going to do is we're going to look at, how do we get the copy, and where is the work site, and how do we construct the parts together to make the product or the movie theater. So there's 2 steps involved. There's transcription and there's translation. Transcription is converting the genetic information or the master copy of instructions into RNA. We call it messenger RNA, so it's the photocopy you make. And then translation is reading this messenger RNA and putting the parts together to structure the protein. So here we see the main piece of DNA, the main set of instructions being copied into a messenger RNA. This messenger RNA will then go out and be read by a ribosome where we can put these parts of amino acids together. So transcription itself is, you have DNA, which is a double helix. And you have this molecule called RNA polymerase. It will bind to an area at the start of a gene called a promoter. It's a promoter because it's like, hey, this is the beginning of a set of instructions in case you need it. RNA polymerase will bind there, unzip the DNA, exposing the template strand, which is the only strand it reads. It doesn't read this other strand called the complementary strand. and it uses this strand to make a copy. And if you look, if there's a T on the complementary strand, it puts an A. If there's a C on the complementary strand, it puts a G. A G on here gets a C. But if there's an A, it puts a U. Well, in DNA, so in DNA itself, A binds to T and C binds to G. But in RNA, what happens is C binds to G, just like here, but A binds to U. Because T's don't exist on RNA. And this is, while there are only 4 different bases in the mRNA, A, C, G, and U, 20 different amino acids can be used in the building of a protein. And so if we look at a section of RNA, let's say that, we have CCG, AAG, CUC, AU, C, and then CCU. These are like words. And this is a codon. So this is one codon here. And this codon corresponds to one of the 20 amino acids. And if we look, this is the genetic code for all organisms on the planet. Every organism uses this code. So for bacteria, UUC on the messenger RNA means that phenylalanine amino acid comes next. And if you notice, there's a couple of specific codons here. There's a start codon, and there's stop, and here's a stop too, and 3 stop codons. So these are the words, and I want to get down here to the picture. Actually, it doesn't really show a nice picture, but this is translation here, where messenger RNA comes out. Messenger RNA will flow through this, this messenger RNA will flow through this ribosome, and it will flow through the ribosome going this way. And in this site here is a codon, and if the codon is here, then this other RNA, transfer RNA, will bring in the proper amino acid. And the amino acid it brings in is based on this. So make sure you read these, get an idea of how translation works. But these are the main players. Well, we've been talking about sickle cell. So, how does the mutation change the protein? Well, if there are mutations or changes in the DNA, right? If you change an A, G, C, or T, then you change what the mRNA is. And if you change the mRNA, then you could change which amino acid comes next. And if you change the amino acid, you can change how that protein functions. And the difference in someone with normal hemoglobin and someone with sickle cell disease is a single amino acid. So I'm going to be posting a couple of videos of transcription and translation and replication for you to watch that I didn't produce. They'll be professionally produced that will be much better describers, and that way you can kind of visually see it. But that is all for this module.